Hi everyone. Today we're going to hear about the neural integrator and the idea that control of maintaining stillness may be a different circuit than the system that allows you to make movements. Our presenter is Aasef Shaikh, who is a chair at Case Western Reserve, and I got to meet him when he was a neurology fellow here at Johns Hopkins. Aasef, thank you so much for being.
Thank you, Reza. Can you guys hear me all right?
Yep.
Great. So thanks for having me. It's always fun talking to you all. Always very thought-provoking discussions. So I'm excited and looking forward to talking more about these things. So the idea, essentially, as Reza said, started when I was back at Hopkins, and I will get into that details. But what we will talk is neural integrators in network model for cervical dystonia. So cervical dystonia is a third most common movement disorder, and it affects about at least 1 million or more people worldwide, and characterized by abnormal twisting and turning of the body part. Historically, people had been thinking more along the lines of involvement of the basal ganglia as a cause for cervical dystonia, but then later on, Buzz Jinnah and Alan Hess, when they were at Hopkins, they started the idea that it could be the cerebellum, which is involved in cervical dystonia. Subsequent studies started looking into involvement of cerebellum, and then more papers came out, more studies came out, and now contemporary view, fast forward 30 years, contemporary view of cervical dystonia, it's neither cerebellum nor basal ganglia, it's a network between multiple nodes of the brain, including proprioception and cerebral cortex. And neural integrator, which is in the brainstem, plays a very fundamental role, very important role. At least we can tell now that in a cervical dystonia, which is most common form of dystonia. So what I will talk today is give you basic concepts of neural integrator, what is the Hopkins connection with the neural integrator, and how the field then moved forward beyond the eye movements into the neck movements, and some contemporary evidence from our work and some other work showing like why we think that neural integrator or the network affecting neural integrator is involved in cervical dystonia. So whenever we talk about any movement, a lot of times we think of precision, and precision is so Mother Nature has designed the homunculus, the picture you can see here. It is designed according to the level of precision that you need for every movement. Like your arms are bigger, your lips are bigger, your tongue is much bigger than the leg and trunk, right? So precision is very important, but simultaneously, we also need to think about persistence. So when you are precisely reaching a place, you want to persist at that position also. So like Reza said, stillness, right? So in order to achieve stillness, you need to put certain parts of the brain in action, and that then assures the persistence or stillness, so to speak. So let's take an example of an eye. So if you're gazing to position 1, for example, and then you are turning your eye to position 2. So when you turn your eye to position 2, multiple things happen after the fact that eye has turned to another position. So one thing is the passive pull of the eye muscle, which is going to pull your eyes back towards the center simply because of the elastic force of the muscle, orbital tissue, and mechanical factors within the orbit. And as a result, eyes may tend to turn back towards the center. Now, brain has to assure that eyes stay where it is supposed to stay. In other words, brain has to create a mechanism to counteract this rotation force that is because of orbit, and that we think is because of the neural integrator. So the idea came, of course, from Wilmer, and a lot of you probably may know this picture. It's a very popular picture. You can see David Robinson here putting search call in somebody's eyes, and in the background you can see a young man who is now the David Z. So what Robinson proposed in contemporary literature in 70s is that there are some neurons in the brain. They know how to do calculus. They know how to do neural integration, how they can take the velocity signal in the brain and convert that velocity signal into position. So these cells have an ability to convert velocity into position. And very simple box diagram here, like this is neural integrator, which is the blue. It receives the pulse of signal and it converts that pulse of signal into a step. This is a classic neuron recording from monkey, a vertical and torsional neural integrator activity. What you will hear and see in this video when I play is that this bright dot will move in the screen, and as the bright dot moves in the screen, you will hear different amount of firing. It'll be like humming bee, like it sounds like almost like a bee. I don't know how many of you, probably some of you already have recorded or record from monkey brainstem, so you may know this already. What you will see is in certain parts of the, when the dot is at certain parts of the screen, the sound will be different, will be more high intensity, higher frequency, and in some it would be quiet. This is the example of neural integrator cell. So this cell receives the pulse of activity neural signal, and then it converts into step, and it changes its firing rate based on the position of the neuron of the eye. And it is not playing. Oh, Reza, would you mind? Can I just try something else to show them the video?
Yeah, of course, up to you.
Yeah, because I really want people to show this video, and I changed it to my work computer and now it is not playing. I'm going to simultaneously open another file where I will show you the videos. I'm going to stop share this, and I will share another one, and then I will go back to this. Sorry, I'm so sorry for that. The computers that I use at the VA, they don't let me go on Zoom. So, you know, I have to change it to different computers. Give me 30 seconds. I'm gonna just okay. You can see this now, right?
Yes.
Okay, so here. Hopefully it will play now. Oh. Same problem. I'm having problem. Even my other file is not playing. Okay, you will have to just take my words for it. So basically, can you see this now or no? No. I'm so sorry. Luckily, there are not a lot of videos, and you're all engineers, so you will understand the trace, so we should be okay. So basically what happens that when, like I said, when this dot moves to different position in the screen, the firing rate changes, and that's the function of the neural integrator, right? So what Robinson proposed is any neural, or he found, is that any neural integrator in the brain is not just perfect, and you all know that. So it requires some sort of feedback, just like life, right? It's not perfect. You need feedback. And for neural integrator, for eye movements, it receives feedback at least from 3 different places. Of course, there are more than that, but these 3 are obvious ones that are affected in disease class, and that is cerebellum, visual system, and orbital proprioception. So if you have any problem in any of these, what you will see is you will see malfunction of the neural integrator. And as a result, when you turn your eye to one side, it will drift back to the center. So you will move the eyes to this side, and then that will start drifting back, and then you make correction. And that is what we call in clinical world gaze-evoked nystagmus. Now, I don't know if this will, this worked. So what you see here is that this patient, when she tries to hold her gaze, I'll play that again. When she tries to hold her gaze to the right side, her eyes tend to drift to the center, along with some vertical movements, but a lot to the center. And she kind of corrects it back towards the intended position. Again, when she's on the looking on the right side, it drifts to the center, and she tries to correct it to the intended position. What we call it a gaze-evoked nystagmus. And David Z in 1976, he described in a major paper about oculomotor abnormality in cerebellar ataxia, but part of that paper was the mechanism of gaze-evoked nystagmus based on the experiment that David Robinson did. And ever since, we always think of gaze-evoked nystagmus as a phenomenology that occurs in patients who have abnormal cerebellar function, especially the cerebellar ataxia. Now, if you put a search card in these patients' eyes, and if you measure them, it looks something like this. So in this graph here, the panel A is when the patient is asked to look to the right side with the search card in their eyes. What I'm plotting is eye position, horizontal eye position on y-axis and time on x-axis. So what you see here is eyes are drifting. Up is right, down is left. So eyes are drifting to the left, then it is correcting, then it is drifting, then correction, drift, correction. So you see this classic sawtooth waveform. And the sawtooth waveform has a decaying exponential, and that drift is always towards the center. When eyes are at straight ahead orientation, there are no drifts at all. It is relatively stable. And when eyes are going to the left, you see exactly reversed phenomenon where eyes are drifting to the right and beating to the left, right? So this is a classic features of gaze-evoked nystagmus as a function of abnormal neural integration, often seen in patients with cerebellar dysfunction. The specific characteristics are there are slow drifts of the eyes towards the center, followed by corrective movements like quick phases, this. Then there are drifts are minimal at one position, what we call null. The eye position related to null determines the drift velocity. So the other interesting phenomenon which happens is this drift velocity is maximum when eye is farthest away from the null position, in this case center, and then it starts going less and less and less, and then it reverses the direction. The other interesting thing is that the direction of drift depends on where the eyes are in relation to null. So these are few fundamental rules, so to speak, that we follow when we classify the eye movements in patients, and we can tell that whether this is gaze-evoked nystagmus and mechanism is neural integration or something else. So this is classic features you will see in somebody who has abnormal neural integrator. So this whole neural integrators and eye movements were discovered in 1970s, and Subsequently, for decades, we were all like trained to think of neural integrator connected, very connected with the eye movements, and that's where the field was. And early 2000, you probably know these people, Doug Crawford and Ellie Clare from York. Doug is still at York. Ellie is, I think, in Houston. We were both, Ellie and I were postdocs together with Dora Angelaki at WashU. And Doug, before she came to WashU, Ellie was a PhD student with Doug. When she was in her PhD in York, she injected mucimol in nucleus of Cajal. And that experiment triggered a concept that whether one could apply the concept of neural integrator, which is very popular in eye movements, to the control of head movements. And indeed, what Ellie found is that when you inject mucimol in nucleus of Cajal, you can see the drifts in the head movement, torsional head position, in addition to torsional eye position in the monkey. So she concluded that nucleus of Cajal is the head neural integrator in non-human primates, at least. She published this paper in Science. Subsequently, Doug had 2 or 3 papers, more detailed papers in Journal of Neurophysiology. And that concept was floating around in the field. And of course, Ali and I talked a lot about that because we were doing postdocs together. So these are the type of things we would discuss on dinner, right? When we hang out. So that's the beauty of working in big labs. So this thing, something I knew for quite a long time, and I had always been thinking about it. Then around 2006, I moved to Baltimore working with David Z. And of course, my main project was eye movements, vestibular systems, VOR. But simultaneously, I was introduced to another faculty at that time. He was at Hopkins, Buzz Jinnah. Now he is at Emory. But Buzz was very interested in dystonia. One day I go to Buzz's clinic, and let's hope that this video works. And it does work. Oh, it's not working. I don't know what's wrong, Reza, with these videos. Some of them are working, some of them are. Okay, I will mimic this movement. So you guys can see my head right now? Yeah? Okay. So when I asked this patient to look straight, he had this type of head movement. He would drift in one orientation, and then he would correct in the other, right? And then when he is here, it is relatively stable. When he goes that, he does this. When he's on the opposite side, it becomes much, much wilder. So in other words, he had drift in his head movement towards a position in the space where head was very stable. And that drift direction changes where the head is compared to that null position. So very much like neural integrator failure, what we see always in the eyes for gaze-evoked nystagmus. So I told Buzz that, Buzz, this is gaze-evoked nystagmus of the head. And he said, we don't call head nystagmus. We call this is a popular, very common condition in movement disorder called dystonia. Of course, at that time, I was not a neurologist, so I had no idea what was that. So, but apparently, there was a condition called dystonic tremor, which is often seen in patients who have dystonia, and that dystonic tremor appeared as if they had nystagmus. So that triggered the whole idea that, what that means is that dystonic tremor could be a condition which is because of abnormal function of head neural integrator, which was discovered in monkeys 4 years ago. So we wanted to start our experiments in that direction. So what we did is we measured 3-dimensional head movements in patients with cervical dystonia. So we recruited about 20 patients from movement disorder clinic. We brought them to the lab. We put a search call on their forehead instead of in the eyes, and we asked them to keep their gaze to the right like eyes are always with the head. And then we ask them to move the head and keep the head on a target, laser target, which is 10 degree, 20 degree, 30 degree to the right, same thing to the left, up and down, and basically measured the head position at different eccentricities. Now, when we do that, when patient was asked to look 30 degree to the right side, for example, what we saw, again, this is head position from search coil. Y-axis is position, x-axis is time. There are drifts in the head position, correction, drifts again, correction, drifts again, correction. So, like nystagmus. On top of that, you see this sinusoidal modulation, which is a different phenomenology, which is a head tremor, which is commonly seen in patients with dystonia. I won't talk about it today because it's a different topic altogether. But on top of that, we see these jerky oscillations of the head, which was very interesting. Same patients, when they were asked to look straight ahead, they still had these drifts, but they were much less compared to the right. And when the patient looked 20 degrees to the left, it was stable. I don't show this here, but when that patient was asked to look further to the left, the drift direction reversed. There were some examples of cases where the null was closer to the center, and in those cases, we could clearly see the reversal. So basically, the head oscillation, jerky head oscillations in cervical dystonia patients followed all the characteristics that we would see in gaze-evoked nystagmus of the eyes in patients with cerebellar dysfunction. What does that mean? That tells us that it is possible that neural integrator dysfunction and head tremor or a head dystonic tremor of the head in cervical dystonia could be from dysfunction of the head neural integrator. So subsequently, what we wanted to do is to really, in ideal world, to characterize the activity of midbrain neuron in patients with cervical dystonia. Now, that is a difficult task because you can't really go with a single unit electrode in human's brain and record the activity. It's like difficult, right? But somebody has done that in 1972. So before botulinum toxins were invented and before DBS was invented, one of the common treatment for torticollis, which is cervical dystonia, was lesioning procedures. And this was lesioning of interstitious nucleus of Cajal. And people in Japan used to do that, a lot of those procedures. Germany, it was very popular. And Russia, it was very popular. At least there were papers from these countries in history. In Japan, they were actually using it to treat seesaw nystagmus also, which we also think could be related to nucleus of Cajal. So these lesioning procedures were very common. And they would lesion the interstitious nucleus of Cajal to treat cervical dystonia, which is interesting. But at the same time, it kind of speaks towards the proof of concept that neural integrator dysfunction and its connection with the nucleus of Cajal and its connection with cervical dystonia. But luckily, I went to a meeting in 2010, 2011, and I met a gentleman, Alexis Sedo. He worked at, or he works at Institute of Chemical Physics in Moscow. And he is my generation, but the lady who he did his postdoc with used to do physiology recordings from human brains with surgeons when they used to ablate the nucleus of Cajal back in 70s and early 80s. So he had this historic database of nucleus of Cajal ablations in 17 patients with cervical dystonia, which were all saved on magnetic tape, and it was not really. They published that, but not in a way that we wanted to do this. So he had the access. So what Alexi and I started doing is started reanalyzing all the magnetic tape data that he had. And the magnetic tape had single unit recording done with the activity of 52 neurons. And we could go back, reanalyze, re-isolate, and reanalyze them. These neurons comprised of eye-only nucleus of Cajal neurons. As you know, in nucleus of Cajal, there is eye-only neuron, head-only neuron, and eye-head neurons. So they had eye-only INC neurons, they had head-only INC neurons, and they had eye-head INC neurons. So it was pretty consistent with what we find in non-human primates. And he also had simultaneously recorded neck muscle EMG, which is very important because as we record the neuronal activity, we also want to know what is happening with the neck. So he had neck muscle EMG simultaneously recorded, and he had electrooculography simultaneously recorded. So we had all the data available in the tape. It also had a voice command. So we also knew what they were talking in the OR at the time in 70s and 80s when they were recording. So we pretty much had everything that we need to redo the entire experiment. So we launched this experiment with our contemporary concept that whether neural integrator dysfunction in cervical dystonia leads to, or head tremor in cervical dystonia is because of neural integrator dysfunction or not, right? We had all the tools that we need to test that question. So our hypothesis was nucleus of Cajal neurons will have impaired signal of neural integration with the head movement. And when you look at the decay time constant of neuronal firing, that time constant will tell us their integrative behavior. So you ask patient to do a movement of a shoulder, for example, because their head is restrained in the surgical frame. So you ask them to shrug their shoulder, and in response to shrug shoulder, like how much is the drift in the shoulder and how that drift correlates with the drift in the nucleus in the neuronal firing. So you plot the IFR and then you calculate the decay time constant of the neuron. If INC neurons have a signal for normal integration, then this time constant will be pretty prolonged. It would be pretty much normal. But if they have abnormal signal, then there will be a rapid decay and it will be a short time constant, right? So time constant was one of the very important parameters we focused on answering our question. And the other thing that we see in patients with cervical dystonia is that their eye movements are absolutely normal. So time constant for the head sensitive neurons would be much smaller, much shorter compared to time constant of the eye sensitive neurons. So we separated the two and we analyzed them separately. I wish that this video plays because this is really cool. This is the eye movement recorded from a human cervical dystonia patient as he's asked to look up or down. I will send you these clips. This is so cool. So basically, you can actually even see here. So this is neuronal firing rate, okay? And they are asked to look up. You see, this is a voice command. It says up. And this is a voice command, it says down, right? So they are asked to look up and there is a robust increase in the firing rate. It more or less persists here. And when they're asked to look down, it is much quieter. So this makes a lot of sense when I play this video and play the other monkey video simultaneously, but unfortunately, none of them are working. If we have time after the talk, I will just go to the clips and show you guys. But what you can see here is that at neutral position, there is some firing rate. When it is looking up, there is an increase in firing rate. When it is looking down, there is a reduction in the firing rate compared to neutral, which is pretty classic neural integrator behavior that we find in eye-sensitive neuron. Now, when we record from the same area, but the head-only neuron, when shoulder is shrugged up, there is an increase in the firing rate, but it decays off to the baseline very rapidly. And when it is down, it reduces the firing rate, but again, it builds up pretty pretty quickly, as you can see from this diagram. So what it means is that neuron is unable to maintain the current or expected desired firing rate, and it decays with much shorter time constant. So we measured them in all the neurons and made few key prediction. So if impairment is intrinsic to the head movement sensitive neurons of the neural integrator, like this. So if neural integrator is abnormal, impairment is not intrinsic to the neural integrator, but is in the feedback to the neural integrator. So in other words, there is something wrong in the feedback here. So one or other, either way, you will see the decay in the neuron, right? So in order to answer these two questions, what you can do is you can record the activity from, one second, which, oh yeah, this is correct. You can record the activity from eye head sensitive neurons, which actually has both, right? So the question is, if problem is intrinsic to the neuron, then if you take eye head sensitive neuron, and if you ask this person to look up or down, that neuron will work abnormally, right? If the problem is with neuron. And if you ask person to shrug the shoulder, neuron will act abnormally, right? But if the problem is in the feedback, then if you ask the person to look up or down, neuron will have normal firing pattern. But if you ask shrug shoulder, it will decay, right? So that will separate the source in the feedback versus source in the neuron itself. So here are the possibilities, right? So if it is in the neuron, then eye and head both will have decay, like activating neuron with eye and head both will have decay. If it is in the feedback, then eye will be normal and head will have a shorter time constant. So this is one eye head neuron. I'm just going to show you one example. So this is the neuronal firing rate. Okay, this is a raster diagram. And in response to head movement, as you can see, the firing rate decays with an exponential fit, right? And the same neuron, when they're asked to make the eye movement, it is more or less staying steady, right? So this is the same neuron which is normally acting in response to eye movement, but not so in response to head movement. And when we had several neurons like this, and when we compare their time constant, eye time constant on most cases is relatively much prolonged compared to the head. Again, this is single neuron, so you won't be able to expect fully normal time constant because it's an ensemble network behavior, right? But the bottom line is it's still much prolonged compared to the head. So what that means is that cervical dystonia, at least the abnormal neuronal activity in nucleus of Cajal in cervical dystonia, is not because of impairment in head movement related neural integrator, but it is because of the feedback that it receives. So it looks something like this now, right? So you have head neural integrator, you get feedback from neck proprioception, cerebellum, and basal ganglia. If anything is wrong in this feedback, then you will have abnormal neural integration. So the next thing that we asked is we perturbated neck proprioception in normal people, nowhere else but at Hopkins with Z in his lab. So a bunch of normal people sat in a search call system, and we had neck massager, like a high frequency neck vibration, which would vibrate paraspinal muscles in the neck. In other words, we wanted to create noise in the neck proprioception. When we created noise in neck proprioception, we measured their head position, right? So normally what you would expect in normal people is you are looking straight ahead,
Aasef, we lost your audio. Oh, Aasef is gone. Sorry, everyone. Looks like Aasef was having some technical problems, but he's back.
Can you hear me now?
Yeah, we can hear you, Aasef.
Yeah, I don't know what happened. Something got disconnected. I don't know where I was last time when I stopped talking. So I will just show you the slide and please tell me if
Neck muscle vibration.
muscle vibration in normal people, right?
Yeah.
Okay. So basically what we did an experiment. You can see my screen, right?
Yes.
Yeah. So basically we did an experiment in search coil in normal people. When you are not vibrating, if you ask them to look straight ahead, their head would be pretty steady to the right, straight, left, straight, right? When you give them a visual target, it's pretty stable. But what we predicted that when you vibrate their neck with the muscle vibrator, like high frequency vibrator, in other words, if you create a noise into the proprioceptive system, then it would have some difficulties in maintaining head, and hypothesis was it would create gaze-evoked nystagmus for the head. So in the search coil, what we did is we had a head fixed laser, which was like kind of a band, and there was a laser coming out of it. And then there was an LED target. I don't know if that room 2 still exists over there, but they had a big screen with an LED targets at multiple locations, and target would line up at different eccentricities, and their goal was to align the head fixed target with the space fixed target, and do that when vibration is off and vibration is on. So when vibration is off, when everything is normal, head is steady. But when we turned on the vibrator, we saw this type of head movement. They align it with the laser, but then they kind of drift back and then they align again, almost like dystonic neck tremor. Now, when we measure it with search coils, so if you look on the left side here, again, these are normal people with neck vibration on, measuring their head position. This is position and this is time. And as you can see, there is a sawtooth waveform pretty much consistent with gaze-evoked nystagmus or dystonic tremor. And when they look to the right, it is drifting to the left, and when they look to the left, it drifts to the right. And when you measure the drift velocity, these are different colors, are different people, and the drift velocity in normal people, it kind of decays over time, and then it is none at 0, and then it reverses its direction when it goes to the opposite direction. So the bottom line is when you create a noise into the proprioceptive system, you are making the head neural integrator in normal people leaky, and they behave almost like cervical dystonia. So then the second hypothesis is all 3 sources of feedback are internally connected. So if you do something in neck proprioception, that then could reflect in cerebellum, that would then reflect in basal ganglia, and it will ultimately change the complete output of the network of feedback to the integrator. So it's not just each individually connected, but they are also connected with each other. So what we did next is that we went intraoperatively in patients. Now this is in current date. These are the experiments we do. And in the OR, we record from their basal ganglia and see whether cervical dystonia patients' basal ganglia, especially the GPI, where we implant the electrodes, whether those cells have any characteristics of integrator failure. Because if that's the case, and if basal ganglia is connected with the neural integrator, then you will see the reflection of that into the feedback also. So these are like three different neurons. The panel A is the GPI neuron, panel B is GPE neuron. GPI is globus pallidum internus, which is the output zone of the basal ganglia. GPE is globus pallidum externa, which projects to GPI, and INC is nucleus of Cajal neuron, which is our neural integrator neuron. Now, INC neuron is not from the same patient as GPI and GPE. It is just showing here from the historic database for just the comparison. What you see in the gray is the single unit activity, and red is the EMG activity. So what you can see is that patient is asked to shrug the shoulder, EMG activity changes, and then it decays. And the decay parallels the decay of the GPI neuron, decay parallels the decay of the GPE neuron, and similar thing was also seen in the nucleus of Cajal. When we measure the time constant of electromyography and the single unit, it is almost in the ballpark. It is not perfectly aligned, but again, these are like EMG reflects the ensemble response, while single unit is single unit, but they are in the similar ballpark in the range of about 2-second time constant. And it is seen here that GPI, GPE, INC, and EMG, they are all have somewhat similar decay time constant. What that tells us that decay that we see in the EMG, the same type of decay was seen in INC, nucleus of Cajal, in integrator cells, and the same type of decay is also observed in GPI. So what that tells us is that gives you proof of this circuit again, that neck proprioception and basal ganglia are connected. They are part of the one circuit, and you can see the exact problems that you see in the EMG or in INC. You see the similar type of features into the tonic neurons of the basal ganglia. And then what we did is intraoperatively, we vibrated patient's neck muscle. As we are vibrating patient's neck muscle, we recorded from basal ganglia. So now the question is that how really neck proprioception changes the basal ganglia activity pattern. First of all, oh, we are still here, right? I just lost you guys. I just wanted to make sure.
Yep, you're here.
Because your picture disappeared, so now I'm just little burnt ones. So okay, sorry. So what we wanted to do is we wanted to vibrate, and as we are vibrating, we wanted to record from the basal ganglia activity. So we did that. The paper is currently in review, but in that paper, what we did is we had 17 patients with cervical dystonia. About I think 10 of them had isolated cervical dystonia, and 7 had cervical dystonia as part of generalized dystonia. So it's a little bit of a different clinical phenomenology, but all of them had neck dystonia. We could record 113 neurons from globus pallidum, somewhere from GPI and somewhere from GPE. And of course, it maintained the physiological distribution. So we had a lot of burst neurons, as we expect in pallidum. There were several pause neurons, but very few tonic neurons. And this is pretty classic distribution that we typically see in pallidum when in cervical dystonia. Now what happened, we record the same neuron, firing rate or activity of the same neuron when vibration is on and when vibration is off, and we compare different parameters. We had about 20 different parameters for burst and pause, and they all measured different characteristics. To summarize, one was firing rate, which is the gain. The second was firing pattern, and firing pattern was then measured with several different parameters, including coefficient of variance, irregularity in the firing rate, how dense is the burst, how far apart is the burst, and whatnot. What was changed instantaneously in response to vibration was the frequency variance. So what you're seeing here is in C, it is GPI cells, and D is GPE cells. As you can see, we had very few GPE cells because we don't really map GPE that extensively, because the goal in OR is to really map GPI, because that's where we are placing the electrode. So we had more GPI cells compared to GPE cells. And we had two different types of cells. One type of cells were responsive to neck movements, and the other types of cells were not responsive to neck movements. So in OR, we find the cell, we ask patient to shrug the shoulder, we see the responsivity to the neck movements. And if we see the responsivity, that is a neck movement sensitive neuron. If we don't, then it is not responsive to that muscle. That's simple. Black is a focal cervical dystonia, isolated neck dystonia, and gray are dystonia, which is CD with generalized dystonia, right? What we see is in presence of neck vibration, the frequency variance of all neck movement sensitive neuron in focal cervical dystonia, most of the times it dropped down. So it basically, neck vibration decreased the frequency variance, making the firing rate more regular, which is pretty consistent with what we see is like undoing some of the bad things that cervical dystonia did, in other words. So it makes it more regular. And interestingly, these are the non-neck movement sensitive neuron. And what you see is in non-neck movement sensitive neuron, in those who have generalized dystonia, there was a lot of those neurons actually reduced their firing activity, as you can see here. Very few were unchanged. While in non-neck movement sensitive neuron in focal cervical dystonia did not have any change, which is expected. But the interesting thing was those cells which were not supposed to respond to neck did respond to vibration. So it was a little flipped. And we have theories for that. We think it is because of the cross-coupling, mirroring movement that we see in cervical dystonia. And the mirroring movement is not just in the efferents, but it is also connected to the afferent. And that's my speculation for this phenomenology. But the bottom line is that when you vibrate a neck, you see instantaneous change in the firing pattern of the network, which is, and that network then projects to the nucleus of Cajal in big picture, right? And then what we wanted to do is we also wanted to see what happened to the ensemble responses. So we were able to record local field potentials from the similar areas where we recorded the single units in the OR. And again, like the dark so above is pure cervical dystonia, below is cervical dystonia with generalized dystonia. And what we found in pure cervical dystonia in neck sensitive areas, as you can see with the black circles here, there was an increase in the field potential in the alpha band. In GPI. In GPE, there was no change. And again, there are reasons for that, and I have theories for that. But in GPI, there was an increase in the field potentials in alpha band. And in contrast, in cervical dystonia with generalized dystonia, there was an increase in neck sensitive areas in the theta bands, right? So neck vibration changed the alpha band activity in cervical dystonia and theta band activity in cervical dystonia with generalized dystonia. And again, like what I wanted to say here is you see multiple different field activities here, like different types of neurons. And it turns out that when there was an increase in the field activity, those areas had very predominant proportion of burst neurons compared to the pause neurons. There were always more burst neurons. And when there was decrease or no change, burst and pause neurons were about the same. And that kind of makes sense with the dystonia pathophysiology, because in dystonia, there are more bursting pattern than leads to dystonia. So if there is more bursting pattern, then they are more likely to make significant change into with the proprioceptive modulation. So that was the connection for that. So the question is that why and what can we make out of change in the alpha band in pure cervical dystonia and change in theta band of generalized dystonia and cervical dystonia. So Andrea Kuhn from Germany, a while back, she did a study where they basically recorded the local field potential from the implanted leads, and simultaneously they did magnetoencephalography and looked at the coherence pattern. And what they found is that this is a picture from their paper. What they found is that pallidotemporal source is theta band coherence, pallidocerebellar source is alpha band coherence, and corticopallidal source is beta band coherence. So what we find in pure cervical dystonia, isolated cervical dystonia, we see the change in the alpha band coherence, and in generalized dystonia with cervical dystonia, we see the change in theta band coherence. What that means is that there is a pallidocerebellar connectivity is disrupted in patients with isolated focal cervical dystonia, which is pretty much in line with Buzz and Alan and many other colleagues in my field have proposed. And what now, after this study, even more so, we think that isolated cervical dystonia is really a different entity and different pathophysiology compared to generalized dystonia, because we are even seeing different coherence pattern in response to physiological intervention. So at least for cervical dystonia, we can tell that neck vibration alters the GPI activity and it changes the pallidocerebellar connectivity pattern in this manner. So take-home message, basically neural integrator for head movement receive feedback from basal ganglia, cerebellum, and proprioception. When there is an abnormality in this feedback, you can see drifts, which can then manifest as dystonia or dystonic tremor. The source of feedback are interconnected. If you change input to or change one source of feedback, that counterintuitively changes the output from the other source, which is actually opening up a very generous therapeutic window. For example, you can treat dystonia by giving botulinum toxin injection, which could potentially change the proprioception by changing the muscle spindles, right? And at the same time, in some patients, you can do pallidal DBS. You see the same effect. You treat dystonia by injecting Botox, or you treat dystonia by doing pallidal DBS. It's very interesting. It's not magic. It's just because you are tuning the same circuit just from a different routes. Again, it opens up two new eras. Like you can even go to cerebellum in those who have abnormal basal ganglia or strokes in basal ganglia. You can go to cerebellum and you can modulate cerebellum to treat dystonia, which is not done currently, but this is something one could explore. And of course, neck vibration can be another thing one could do for rehab. So with that, thank you. And sorry for all the technical glitches.
That was great, Aasef. Thank you so much for very slowly walking us through the procedures. I was going to begin the question by asking you, since Doug Crawford's work in 1992 or 30 years ago, have there been any development of any animal models that have looked at the role of the interstitial nucleus of Cajal for and its function in the integrator or any other components of the circuit?
So Reza, the work I think he did in 1990s with Tudis Willis for his PhD, he basically focused only in eye movements in 90s. And the work he did with Ali in 2002 was about the head movement. But I don't think that anybody I haven't come across any monkey model for cervical dystonia. Of course, we do have mice model, like genetic mutants for dystonia, but we don't have any non-human primate models for cervical dystonia. Doug says that his model is pretty much the model for cervical dystonia. I mean, you can call it a model for cervical dystonia, but it's really not, because what his model is lesioning nucleus of Cajal, and that's not really what happens in cervical dystonia. We think that it is the way proprioceptive signal is handled that is abnormal, right? And the idea is probably there is something wrong with the cerebellum, because Buzz has shown Purkinje cell dropouts in autopsy tissues in humans. Of course, it's very non-specific and from many different locations in cerebellum, but still, it is there. My best take on all this is problem is not nucleus of Cajal, problem is not the proprioception, problem is somewhere in between where proprioception goes into the nucleus of Cajal, and that is either cerebellum or basal ganglia. Now, basal ganglia activity is abnormal, as we can measure, right, intraoperatively, but is it primary or it is secondary? Very likely it is secondary, because we did a study we published in Neurobiology of Disease in 2019, I believe, or 18. There we did more detailed analysis about Cajal versus, and most of the cells that we found on basal ganglia were leading neurons, not the lagging neurons. And the idea is that you cannot have driving neurons abnormal because of proprioception. It did not fit with quite the theory, something wrong in basal ganglia. So again, it comes down to cerebellum. There is something wrong in cerebellum, and it is just making the proprioceptive signal noisy. So that is an open for investigation. If somebody can record from human cerebellum in dystonia.
Are there, let's see if there are other questions for
one more thing I must say. Roy spoke with you guys, right?
Yes.
Yeah. And did they show the data from their lab? Like when we invited him here, he had very cool optogenetic modulation of deep cerebellar nuclei, dentate, in mice models. And what he found was if you do rhythmic modulation, it will lead tremor in mouse. But if you do the bursty modulation in dentate, that would lead dystonia, okay? And what we find in cervical dystonia is more bursty pattern in GPI. So it fits with what Roy had modulated and created dystonia. We see that pattern in basal ganglia. So that tells me again that there is something wrong in the cerebellum.
Yeah. Any questions for Aasef?
Yes.
Yeah, so thank you for the clear talk. So I think I'm confusing like vertical and horizontal component. Like for the Cajal, my like limited understanding, it's more vertical.
Yes, you're right. You're right. So it's Cajal is vertical and torsional, even for the head, and for the horizontal, we think it is either nucleus of Darkenschweiss or the perirubral regions in the brainstem. And I do have proof of principle, at least in humans. I had a patient where there was a wrong place electrode, not my patient, somebody else's patient. And I was interrogating that DBS electrode, and it was very deeply placed, so it was almost near red nucleus. And when I increased the charge from the distal lead, it actually led to the head turning. So that tells you that it is in the that it can happen with, if you overcharge the integrator on one side, it can lead to head turning, almost like a relative lesioning, right? So we think that horizontal is perirubral in humans, and vertical and torsional is still in nucleus of Cajal. But remember, we do shoulder shrugs in the OR. We can do head turning in horizontal, right? So these are like roll movements. Because in OR, unfortunately, we can't have them do horizontal movements because they are fixed.
Yeah. Yeah, thank you.
Yeah. Reza, any ideas? How can we move this forward?
Well, I think your focus on the cerebellum sounds right to me as well, and I would guess that it's something to do maybe with the flocculus, something down there that may be an issue. But yeah, it really, like you say, it really needs an animal model. And Roy Silito's work and Amanda Brown's work, and those individuals can hopefully move us forward.
Mm-hmm. Yeah.
Are there any other questions for Aasef? Okay, Aasef, thank you so much. Have a wonderful Tuesday, everyone.
Thank you. Hey, Reza, I want to talk with you. Can I call you on your phone for a second?
Yes.